Oh, good morning.
Good morning, everyone. Um, Great. So I think I will just share my screen. Okay. So I'm guessing everybody is able to see my screen.
Yes, Tina.
Okay, great, great. How are things? How is everyone doing with the what do you call it? With the I haven't gone over that Trudy Schubach's assignment yet. I have just not had the time. But over the weekend is what I will go over them. Um, I just went through a few, but I will get back to every one of each assignment with a response by the weekend. I think that's only when I'll get some time. Um, but apart from that, I think last class we discussed more or less the places where it was difficult. I still need to get a better handle on images that can that can help Anandita's bit. But apart from that, most of the most of the parts which was difficult in the paper, um, the dorsal ventral polarity paper, I think we kind of figured out how to go about them, right? So the assignment should be kind of uh, uh, easier to achieve. So I'm expecting by the weekend, you guys will be able to submit that assignment as well. If there are any further uh, areas where you find difficulty understanding the paper, you can tell me now, and we can see how we can help that. So, is there anybody with any more aspects that are difficult in that paper? Okay, great. So the silence is super motivating. Okay, so essentially, we're kind of getting done with the the idea of how Drosophila is developing, because I think um, what um, what I need to now be able to just explain is how complete the explanation for is how this one embryo, which is multicellular, is getting to look like this Drosophila larvae, and put in every information into this larvae so that it becomes into an adult. We've gone over um, we've gone over um, the concept of utilizing gradients in the first half of development, that two opposing gradients can get the ball running. So gradients of bicoid and caudal or nanos, one from anterior and the other one from posterior, is going to set up the first set of genes, and those expression of the first set of genes are going to divide the embryo into four broad stripes. So essentially, what is this latter half post gradient all the way to? I mean, we keep calling it segmentation. Essentially, two things are happening here. The two things that are happening here is that they need to be divided into repeating units. Two things that need to be achieved by the end of embryogenesis. After that, everything is easier for the animal to go about. So one thing is the division of these group of cells into segments that will define, okay, this segment is head, this is thorax, this is abdomen, this is tail. So first thing first is to divide the zones in proportions that would give rise to segments of head, thorax, abdomen, tail. Fine. One thing is division of the embryo. And the second thing is telling that now that you've divided, you have to have a certain identity and maintain yourself within this identity as long as the animal exists. So you're not going to change your identity. So it is specific, it is essentially giving rise to this whole thing of specification and then determination. So without, basically this is what the this whole process of segmentation is about. And bicoid and nanos, the gradients that set up, are two essentially starting points that will result in the result in the achievement of these 2 points. So first, they will have specification of the different specification of each of these cells based on their transcription factors or the different zones that they get divided into. And then once they've been divided, they're going to be determined. For what structures they're going to develop. So now you can essentially imagine that within the head region that gets specified, you will have determination of structures that are going to give rise to the eye, the antenna, the proboscis, and so the head is going to have structures that will now be determined. The same is true for the thorax, abdomen, and the posterior tail region. So now the thorax will have different segments divided further into, that's the part of determination. And each of the different T1, T2, T3 parts of the thorax are not going to be the same, because from the first half of the thorax, no legs will come, but the second one will give rise to legs. The third one will give rise to legs and a pair of wings. So they will have identity of thorax, but they will have determination of different structures that they're going to give rise to. So once this is done, the job of development is done. Then the animal just needs to develop like that, because the embryo would have laid in all the information. Now it's like an origami that is going to set in, because the marks where the folds have to happen have been set in. So you can imagine this is like an origami, game of origami, where you have a sheet of paper where all the folding marks are present. The embryo does that. These different genes that are going to be establishing all of this are there. And wherever the folds have to be made, they're marked. The steps are marked: number 1 fold, number 2 fold, number 3 fold, number 5 fold. And as a result, now the developing animal knows this is what the series of events is, and I'm going to just follow these rules, right? So essentially, this is the process that takes over from the reading of the 2 opposing gradients. Does that make sense to everyone?
Yeah.
Okay. So now, so what I just said is essentially all of this. So this is the embryo. This is how it will, this is the multicellular, like cellularized one. But when segmentation happens, post-gastrulation, you have all these different segments forming. And then these different segments, as they form, they're going to develop. So this is the segmentation event. And as the segmentation event happens in the adult, in the first instar larvae, we're going to see the outcomes of how it changes the banding pattern on the cuticle. So depending on how the cuticle looks of a first instar larvae, you can actually discern which are the different segments that have been missed, because essentially all of these bands come from different segments of the embryo. Now, this was the simple principle. This was the principle that was utilized for the screen, and yeah, I think we discussed that. I'll come to parasegments in a bit. Yeah, so the screen done by Nuslim Follard and Eric Wieschaus resulted in 3 different types of mutants that they got. So one of the mutants, they found these 150 different mutants that affected. So what they were doing essentially is scoring these cuticle phenotypes. Now this is a wild type first instar larvae with all the different cuticle, with the different 8 different segments, abdominal segments and 3 thoracic segments. So essentially, if you can see the
Tina, I think we're seeing another screen. I don't think we're seeing the slide you're showing.
Oh, really? I've been talking to the wrong slide.
We can see the segment polarity mutant screen. I mean, the slide now.
You see this?
Yeah.
Yeah. Oh, you see this. Now segment polarity, goosebump and all of that. Okay, fine. I want to show you another slide. Thanks for bringing it up. Stop share. Share screen. Their bioelectric. What do you see now?
We can see the segmental phenotypes by Volhard and Weishaus.
Oh, okay, that's a slide. Fine, great. Okay, thanks, man. Who was this?
This is Ron.
Oh, Ron. Hi, Ron.
Hi.
Before, have we?
Sorry, yeah, I think in one of the earlier classes.
We did. Okay. Ron. So you're a undergrad or a PhD?
No, I just joined as a PhD student. I mean, I'm yet to come there.
Yeah. So you're a 2020 PhD student, huh?
Yeah.
Okay, great. Thanks, Ron. Okay, so, I mean, essentially what I was talking to was this slide in the, I don't know which slide you guys are seeing, but this is the slide that I was showing and speaking. So essentially what I meant was about this embryo and the point of segmentation. So I'm not going to repeat everything I said, but essentially the embryo is going to be dividing itself into broad segments and then determining each. So it's first specification and then determination. Specification of each of the, specification means division of this whole group of cells into head, thorax, abdomen, tail. That is specification. Then determination to give rise to individual structures that those segments are going to be forming. So it is important for maintaining identity and what I'm going to become. I'm going to become an eye. First of all, I'm part of a head structure. Second, within the head region, I'm going to become the eye, or I'm going to become the antenna, or I'm going to give rise to proboscis. So this is what segmentation and this is what the process of segmentation is going to achieve. Okay, and then essentially you will have a first instar larvae, which will look like this. And yeah, so coming to the embryo, the different segments. So, and then within the segments, you have different structures that are going to be defined. Now, each of these different thoracic segments or abdominal segments will have different identity. And they're not the same, although they're abdominal segments, they are not going to give rise to the same structures. So that information is loaded in each one of them. And finally, each of them know what they have to be and what they have to give rise to. So this is now a job done in the process of at least establishing what you guys are going to become. And then the animal as a larvae, once it ecloses as a first instar larvae, then starts developing on that mode. So the whole origami example is what I meant here, is to say that, yeah, now the animal knows where the folds are and I have to fold like this, fold like that, and give rise to an adult, give rise to a larvae which has to look like this. So all the information is loaded here, and now the animal needs to just follow the instructions. And following the instructions is the job of this guy. And then development is just a process that will eventually be the outcome of, developing as a larvae and then developing as an adult fly is just the outcome of that process. Now, bicoid, nanos, and all the other gap pair rule genes that I just mentioned in the last class are like those, it's like drawing the folds on the sheet of origami paper so that one can read it and then follow. But how they should be drawn, where they should be drawn, which should come first, which should come second, that's what the two gradients are going to, the two gradients and the following steps are going to lay out. Does that make sense to everyone?
Yes.
Great. Okay, so I will skip that slide because I find that I'll come back to that later. So you can see this. This is a Drosophila larvae, and the anterior and the posterior and abdomen, it's essentially the two broad divisions, anterior, posterior, dorsal, ventral, which I think now everybody should even be very clearly, saying this every class, is done very early. What the subsequent steps happen very early, even during oogenesis, but during embryogenesis is what the divisions, the segmental phenotypes get defined. Now, what Nusslein Follard and Eric Wieschaus did, their question was simple. Their question was, how did the animal get this pattern? How did the animal develop like this? They knew, but till then, Nusslein Follard and the work done by Nusslein Follard, Gary Struhl, Eric Wieschaus, even my PhD supervisor Herbert Jekyll, they had some understanding of anterior posterior axis was, bicoid was known, hunchback was known, and dorsal ventral polarity is established by the mom was known. But how did the animal do the next series of things was not clear. And I think what was very informative at that point of time were some mutants that existed even before the screen was done. And some mutants, I mean, how did they even choose the cuticle? So what Eric Wieschaus and Nusli Fuller did were actually screening for these cuticle patterns. So if you see the cuticle patterns here, these are, so whatever white here is actually hair. So and these denticle belts, they're called denticle belts. And each denticle belt has a typical pattern of these, I mean, they're sensory neurons, but they actually have a hair or a bristle, not called hair, but bristle. So a particular bristle phenotype. Now, this bristle phenotype is not the same from this first abdominal segment to the eighth one. They're not the same. And you can appreciate that this one is like this one is like this. You might find that they look similar here and here, but they're not. And then gradually towards the A6, A7, A8, it looks very different. So essentially, they utilized this bristle pattern phenotype and the differences. I mean, and that there's a repeating pattern. You have a bristle denticle belt, and you have a side which has bristles and then no bristles, bristles then no bristles, and also within the bristles it's not the same. So this was enough as a question to start off with as to how did the animal even achieve this? Because the question was, if I could understand how this animal achieved this, I will be able to understand the mechanisms that even set this pattern up. So this is essentially defining how segments get defined. So that led them to utilizing the cuticle as their question. So the cuticle denticle belt patterns and how they look like what they look, and what defines them was the question that led them to the screen. And 1 second, just a minute. Hello. Yes. 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 Just a minute. 77 20 365 44 790. Okay. TINA M @instem INSTEM INSTEM So essentially what I was trying to, using this dendrical belt patterns, the two people essentially, Nusselt Folard and Eric Wieschaus, set up the screen where they mutagenized flies, mutagenized embryos. And how did they mutagenize and all of that? You should read it. It's very simple again, not rocket science. But just hitting the mute embryo with EMS at a dose that will result, the dose was standardized to be causing mutation in one gene in one animal. So if if the dosage is very high, then multiple mutations can happen. But the dosage of EMS, I think EMS is what it is, was used, and the dosage that was used was standardized so that there was only one hit per genome. And essentially then the consequence of that one hit, one mutant gene, if there was any effect on this patterning, is what was scored. Now, they wanted to particularly ask what is zygotically contributing, because what maternally was contributed was clear at that point of time. Anterior and bicoid nanos, all of that was understood beforehand. But they wanted to ask, what does the zygotic genome do so that will result in this patterning defect? So the screen was designed in a way that would lead to zygotic mutants, and the zygotic mutants, so essentially what the zygotic genome contributes to is what the screen also led to, screen also led to its discovery of that. Okay, and that together with the understanding of what the maternal contribution is, and then with this screen that led to the understanding of what zygotic genome can do, established this whole roadmap of development of this animal. So how maternal genes are being read by the zygotic genome, and then how the zygotic genome is then further setting up the downstream flow of information. Okay, so as they scored these phenotypes, so essentially imagine now you're sitting at the microscope. Every day you have thousands and thousands of cuticle preps that you're doing, and each cuticle is each cuticle in one vial is one phenotype or no phenotype. So they scored many, I can't even tell you, 3,000, 5,000 cuticles, probably undermining the effort, but essentially these two people were sitting at the scope and doing everything. So they would make, they would have the embryonic mutant. So minute the mutation, so the crossing scheme was such that once the eggs were laid, they will collect the cuticle from those eggs. So they would do a cuticle prep, which is not trivial because you have to treat the embryos to obtain the cuticle, and then you mount the cuticle, and then you visualize the cuticle under the microscope. So what these two were doing essentially is mounting cuticles and seeing these patterns. Now, of course, there are many genes that did not affect the pattern, although perhaps there must have been mutants. I mean, of course, there were mutants, but they did not affect any aspect of this segmentation. But then they found mutants that started giving them phenotypes of different kinds. Now you can imagine if these are the kind of things that you're seeing every day appear, you might find this is similar to this and this is different. But what they actually realized is that essentially they got three types of phenotypes. Three types of phenotypes, which meant they could distribute all the different 150. So in the screen, they found 150 mutants. So they could actually distribute these 150 mutants into three groups. So the first group, which I mentioned last class, are the gap mutants. So in them, huge segments were gone. I mean, huge number of segments were gone. So the segments disappeared as blocks. So like here, the entire thoracic segment is gone. And many of the abdominal segments. So from T1 to all the way to A5, A6, A4 is gone. And here, T1 is there, but T2, T3 is gone. But although it looks fine, but if you see this region, it doesn't look the same as the control. So all this is gone in this particular animal. Now here, the middle abdominal segments are gone. So, which means that segments could, it could mean that segments, these mutants had different regions that were missing. But what was also seen in these type of mutants, that together a bunch of segments were gone. They were disappearing together as a block. So meaning whatever the gene was that was mutated in them contributed to the establishment of that particular block. Now it might contribute to some of the thoracic segments or some of the abdominal segments. So this particular mutant contributes to T3 and all the way to A6. So from T3 to all the way to A6 is what this gene contributes to. Whereas this particular gene contributes all the way from T2 to T3. And here, the gene contributes to the entire T1 to T5. No, 5, 6, 7, 8. Yeah, T4, A4. So huge blocks of segments disappeared. These they termed the gap mutants. And so what they identified as going as huge segments was gap. Then there was another set of mutants that they identified. So now you don't get mutants organized like this, but they did this organization after scoring all the different 3,000 different cuticles or 3,000 to 5,000 different cuticle preps that they have done. And after having scored through all the different mutants. So they began to see some kind of coherent language being spoken. Because I'm trying to what I'm trying to get at is trying to appreciate that this is not trivial. To see this and then see this, I don't know if I have pictures of the I don't think I have pictures of the other mutants. Okay, I don't have pictures of the other mutants. Wait, just let me. Yeah, I'll just show you one slide. One slide here. So, does everybody see this slide? Gooseberry wingless. This slide here.
Yes.
Yeah. So I showed you that one slide there, right? Now, what I'm trying to make this point is that they were getting all these kind of different phenotypes, right? Now, these are actually segment polarity mutants. This is how the segment polarity mutants look. So seeing this, they had to make sense out of it. As a person, being a Drosophila person, if you were to look at, if I am looking at it, I will not be able to say the differences immediately. Yes, with more and more of mutants coming up, probably the differences will get better. And it is what I'm trying to say here is a careful bent, the bent of mind to be looking at things with a lot of detail and with clarity in mind and thinking about it is what led them to classify their mutants into 3 different types. Because essentially, once the classification was done, then the understanding of development was done. Because the mutants classified into those 3 different types is what development does. And they could see their phenotypes and do exactly the same distinction that development did. And this is not trivial. So it might look like, oh, the 3 different mutant types, 3 different types of genes, 3 different types of events exist. But those 3 different events, so those 3 different types of events, not just they happen, how they would be linearly placed in the process of development, which one follows which one, do they all happen at the same time? Would these genes, which are gap genes, pair rule genes, or segment polarity, because in their screen, they got these different, they divided them into 3 different segments, 3 different types. But the question then was, okay, fine, then how are they, what is the hierarchy? First of all, is there a hierarchy? And how are the 3 different events talking to each other? The first one, broad segments are missing. The second one, every other segment is missing. And in the third one, which is the segment polarity, there seems to be missing, the segments are there, but either the segments have their front missing or their back missing. So the different, so even if the animal has all the different T3, thoracic and the abdominal segments, they might have their ahead and behind, or the anterior and posterior, mistakenly represented. So this, the clarity that the events are not happening all at the same time, and there is a hierarchy in the events, is also what got established by these 2 people. I mean, this is a lot of thorough and very thorough and deep analysis of their data that they did to get to this conclusion. So, which is why I always appreciate when you look at your data, not to do an experiment, the next one, as the first obvious thing, but to look at the data and to ask the data, what are you trying to tell me? And did I understand everything right? If you have understood everything right, then you pose the next question and ask, okay, if I understood everything right, then my next question should prove me by doing these experiments. And if the epistasis is correct, if I manipulate this step, my subsequent steps should be missing. So however you understand, you set up your next experiment. So the more you understand, the more clarity you get, and a much better experiment gets defined as a consequence. So what these guys did is essentially understood, divided their mutants into the three different three different types, and then also established that these three different events do not happen all at the same time. There is a genetic hierarchy, and this genetic hierarchy is sequential. One is specification, while the other one is more deterministic. Okay, so now does everybody see my original slide again, the gap Newton Schling-Follard slide?
Yeah.
Okay, great. Okay. So now once they had these genes, so what did they see? So the mutants that they call the gap genes, they divided the embryo into these 4 broad domains. And as I mentioned in my last class, the divisions get more and more with the establishment of the pair rule genes. And finally, the segment polarity genes are going to divide it into as 14 stripes. Now, each stripe is going to be a segment. Okay. Now, so it goes this way: specification to determination. So Gruppel is a gap gene, defining the thoracic segments all the way to A5, sorry. So in a Gruppel mutant, all of these were gone. So broad blocks are of this entire block is gone. Now odd-scaped, even-scaped, paired, round. So Gruppel and Knubs are the 2 gap genes that are shown here. And these are the different pair rule genes that are shown here. And you can see that in odd-scaped, each of these odd segments are gone. So 1, 2, the second thing is gone. The third, fourth is gone. So it's called odd-scaped. So every odd segment is there, but the even ones are gone. And the other one is called even-scaped because the odd segments are, however, mixing it up, but whatever is present here is gone there. So the even ones are seen and the odd ones are gone. Now paired, round are different pair rule genes because the gaps and a part of the other segment were gone. So you can see that these are, in my opinion, to me, the pair rule genes would have been the hardest to score. The gaps were probably the easiest because you have large chunks missing. But to me, the pair would have been the hardest, the pair rule genes, because to discern this and that they are different, I think this is quite tough. So these would have been the toughest to understand. Now the segment polarity genes were also okay, easier because they were clean. Because like this one, gooseberry, in between the gaps are missing. So all these denticle belts, hair, the bristles were closer to one another. So essentially they didn't have the gaps that are seen. So that meant that this particular gene was necessary for giving rise to this region which lacked the bristles. But here, unlike the gap genes, the segments are not missing in larger blocks. And this one, unlike the pair rule genes, have all the different segments, but in between each segment, whatever the details are, that is missing. And same is true for patched, the other segment polarity gene. Now, there is a remarkable distinction between the type of genes that were identified between the gap and the pair rule genes versus the segment polarity genes. And that remarkable distinction is that as they were identifying the mutants and they were actually trying to understand what genes they were, what they observed is that within the gap and the pair rule genes, there were no signaling proteins like hedgehog, notch, wingless. So any of these signaling pathways did not appear in this, but they appeared in the segment polarity types. The entire subset of the gap and the pair rule genes are actually all transcription factors. So, meaning there is signaling pathways. So, if I were in the class, I would have asked you why is this, but since so essentially in the gap and the pair rule genes, every gene that they identified was a transcription factor. So it was very cell autonomous. Transcription factors will be within the cell, in the cell, and however that cell reads it, the information is going to be very cell autonomous. And its target gene, if its target gene is a signaling factor, that's a different thing. But the readout of that particular transcription factor is very cell autonomous. So this, if you can then appreciate, would specify what each individual cell has to become. Now, as they read the gap and the pair rule genes, they get specified into head, thorax, abdomen. But now within the head and the thorax, they have to be able to now talk to each other to define, okay, I need to become the eyes, I need to become the proboscis, I need to become the antenna. So now the cells have to cross-talk amongst each other to be able to define duties within the particular segment or within the particular, within the segment. So then is when the utilization of signaling pathways comes into picture very handy. Because what signaling will do, okay, I have even skipped and I have very high even skipped, and I'm going to make a lot of wingless. And as I make wingless, I will release it. And as I release it, my neighboring cells will be sensing wingless. And in those cells, I will block the expression of genes that are going to be necessary for bristle formation. So if they are not bristle, they are going to become this white domain. So in a way, the signaling helps communicate each other, communicate the neighboring cells what your identity is and how you can influence the identity of the neighboring cells. So, and then these cells, since I have very low even skipped, I'm going to make engrailed, and engrailed is going to be sensed, and engrailed cells are going to make hedgehog. Hedgehog is going to be released. I will tell my neighboring cell that I am not a bristle and you become a bristle. So this communication back and forth is what is going to define who is who and establish polarity. So which is why they were given the name segment polarity, because it seemed like in these mutants, some kind of polarity was lost. So now, if this is a particular segment, it has an anterior and a posterior polarity. These mutants, this was lost. And essentially, the entire chunk of the segment polarity genes had some transcription factors, but a majority had these signaling factors like engrailed, wingless, hedgehog. So either signaling ligands or their receptors or their downstream effector molecules. Does that make sense to everyone?
Tina, I just had a doubt.
Yeah.
You're saying that the signaling pathway started after establishment of pair role genes and helped establish the segment polarity?
I mean, that's what the screen said, yeah.
Okay.
Yeah. So if you look at all the genes in the gap and the pair rule and the segment polarity, there is this hierarchy of transcription factors that are there in the two preceding sets. And segment polarity is mostly utilization of some transcription factors, but signaling pathways become very prominent here. Correct? So is that clear? I mean, I don't know if that clarifies your doubt.
Yeah, it does. So basically these transcription factors start acting after the pair role genes are established, right?
Sorry, come again?
So I was just wondering that these transcription factors will start activating, or they're activated after the pair role genes are established, right?
So pair role genes are transcription factors themselves. Gap genes are transcription factors themselves. I mean, I'll probably, wait, just give me a second. Okay, so I think this is what I will probably, we went through this slide last class. So, so these are the, so say these are the three different transcription factors that are there, right? Now, the gap genes, so there are three different gap genes. However, they are red, it'll induce the transcription factor, it'll induce the expression of its downstream effector. So gap gene transcription factors will induce the expression of a pair rule gene, right?
Okay.
So these are the different gap genes, right?
Yeah.
And then how are they red? Their combination will give rise to more gap genes, right? So I told you last class that they get further refined in time, right? Yeah. And okay, we'll not do this. Yeah, sorry. So, however, those, where is my slide? Sorry, I thought I had a better slide than that. Okay, so essentially, this is what probably is. So the gap genes, which are transcription factors, their downstream, however, whichever zone or whichever cell has it, sees it, will then give rise to the main first set of gap gene expression. And then depending on their further refinement, so if this gap gene is going down, this is going down, there is going to be a separate set of information which is going to be red. Now this is going to refine the expression of the gap genes, and these different, so what are the stripes here? Essentially the stripes are downstream target genes that will get activated. So these target genes are nothing but the pair rule genes. So 1, 2, 3, 4, 5, 6, 7. So the expression of, this is the expression of, say, one of the pair rule genes, even skipped or whatever. So however the gap genes are red, these transcription factors, the combination of the transcription factors is going to divide the embryo into the different um, is going to give rise to expression patterns of pair rule genes, which will be seen as these stripes. And now this is probably, and these stripes are going to be what you see as when you see the even skipped stripes. So the even skipped is one of the pair rule genes, which is going to be read through the different combination of the gap genes that are there. Now these stripes are not the stripe that the embryo, this is not segment, this is not dividing the embryo into segments. It's just that genes appear as stripes, which means that there is kind of repetition of these, the even skipped or any of these pair rule genes appear as repeating units. And how they appear as repeating units, I mentioned in my last class, but briefly here. But you can see that however even skipped is, there are all these different gap genes present, giant, Kruppel, Knubs, giant, Hooker-Pine, and this array of gap gene expression is being read to result in the expression of even skipped as these 7 stripes. And each stripe here is a consequence of a different enhancer, which is bound by a different transcription factor. And so this particular enhancer is perhaps controlled by a giant, or this enhancer is controlled by a giant, this is controlled by some other gap gene. So what I'm trying to get at is the both the gap and the pair rule genes are all transcription factors which are going to define the cells, which are going to specify cells in these repeating units. So the cells here will not be the same cells as here. Now, even though this is even skipped is present in all of them, there are other pair rule genes which will not be present in the same pattern. So the expression of pair rule genes that is now going to come up. Okay. So the expression, so now the expression of the pair rule genes, the stripes that you see for even skipped may have further segregation in them depending on the other pair rule genes. And now the combination of the pair rule genes that are there is going to give rise to the segment polarity genes, which are basically these signaling pathways. Does that make sense to you now?
Yeah, it does.
Okay, so essentially the transcription events are preceding the onset of segment polarity genes. Okay, now this is a paired. So how did this even come into picture? What they observed is that in a paired, so this is now establishing the hierarchy that pair rule proteins, so that gap proteins came before the pair rule and pair rule proteins came before the segment polarity. Now so essentially in a pair rule mutant, the segment polarity genes were missing. So if this had been the other way around, you would have seen a segment polarity gene affecting the expression of a pair rule gene. But that wasn't seen. What was seen is that if pair rule genes are mutated, the segment polarity genes were lost. So this is a paired mutant, and this is the wild type expression of wingless. And in a paired mutant, the expression of wingless so however many stripes are here, 14 stripes, and so the number of stripes of wingless expression was missing. Now going back to what paired looks like, I think wherever that picture was. paired. So now this is a paired mutant where each of these segments were missing, and each of these segments missing also what was seen is that not just that these segments were missing, the expression of some of the segment polarity gene like wingless was also missing. So kind of what they also found is that the pair rule mutants, now if you were to look at wingless mutant, paired and wingless mutant will perhaps they'll have features that will be similar. Because if wingless is necessary, paired may have many other phenotypes, but it should recapitulate a part of what wingless phenotypes are. So if a paired mutant, if paired is controlling wingless expression, then if you lose paired, the kind of phenotype that you see will also be recapitulated when you lose wingless. Now if paired is at hierarchically more superior, there'll be more phenotypes that are seen in paired mutant that may not be seen in wingless, but still the phenotypes, there will be phenotypic similarity. Now if this controlled its downstream, if this transcription factor, the target gene is this, wingless, so in a paired mutant, paired and wingless mutants should look similar. So this was the other approach that they used to set up this hierarchy. Does that make sense to everyone? That how the idea of a downstream target gene and the similarity in the phenotypes were also seen in the screen, right?
Yeah.
Okay. Okay, so now essentially this is what, so from the 7 stripes of the pair rule genes, they were red. So each of them was then split into more further divisions, and this would make it into 14 stripes. I mean, because there are 14 striped expression of the segment polarity genes. And so it was gradually being divided from 4 stripes or 3 to 4 stripes of the gap genes. You got 7 pair rule gene expression pattern, and from the 7, the division of expression of the segment polarity gene was seen into 14 different stripes. Now, these 14 different stripes would talk to each other. Now the paired mutants are, if the pair rule genes are mutated, it's not that you don't see any stripes, it's just that you see the 7 stripes, because while this is one pattern of pair rule gene expression, there are the other pair rule genes. So each of them controls one stripe, the 14 stripes that are seen of all the segment polarity gene. These 14 stripes that are seen here is an outcome of reading all the different pair rule protein expression. Yeah. Okay. Now, so essentially, how would now the pair rule genes give rise to the second set of segment polarity genes? So the 14 stripes that you see. So even-scaped, Fushitarazu, odd-scaped, there are so many other pair rule genes. Now imagine all of those pair rule genes are being read simultaneously, and then the embryo is divided, is showing the expression of the segment polarity genes in these 14 different stripes. Engrailed looks like this, wingless looks like this, but they do not overlap, or maybe there is some overlap of some of them, but it's not that the 17 stripes here are essentially the same here. Engrailed, now I will show you this expression here. So engrailed is expressed here, wingless is expressed in non-engrailed cells. So they do not overlap. If you were to do in C2s of these guys together, you will see it is engrailed, wingless, engrailed, wingless. So they are adjoining each other. They're not in the same cells. Some of them may be in the same cells, but engrailed and wingless are not. Okay, now the job of the segment polarity genes is to, all of this is in a way dividing the embryo into segments. Now, the job of the segment polarity gene is to give them identity. Now, how is this happening? So, okay, now I will move to another slide here. Okay, does everyone, is everyone seeing my slide, which is maternal gradients, gap domains, and all of that?
Yes, Tina.
Okay, great. So this is essentially what I've been trying to blabber here. Now see it for yourself and understand it for yourself. The maternal gradients, red, and this undulating pattern and repeating pattern of the gap domains, these are further read and further defined into alternating patterns of the pair rule genes. And you can see a new pair rule gene, they're alternating. So it's even skipped and Fushitarazu. Now they're further read by the segment polarity genes. Now the cells that see even skipped and cells that do not see even skipped will have different identity or different polarity. So the ones that see highest even skipped will become engrailed positive cells, and the cells that do not see even skipped become wingless positive cells. So this is how this particular stripe of even skipped is going to give rise to 14 further segments. Now these are called parasegments. We don't call them segments, essentially the main segments, they are parasegments. And these are the boundary between the engrailed and wingless positive cell is called a parasegment boundary. So this is a parasegment. All those 14 stripes that I'm talking about, they're essentially segmenting the embryo, but not in terms of the real segments. They're parasegments, because now from the parasegments, the anterior and the posterior polarity that is now going to be generated is going to be defining the actual segments that the embryo is going to develop. Now, there's called segment polarity because essentially for this reason, that the sharp boundary that it creates, there is one wingless positive region and a wingless negative, but an engrailed positive region. The cells do not mix. There is something that there is a clear demarcation of this boundary, and the cells respect this boundary. They will never interfere with each other's, they will not go and mix in between. So there is no mixing of cells, of engrailed positive cells and migrating into wingless, as if they have marked their territory and they respect these boundaries. So the way they appear defines anterior and posterior part of the segment. So there is polarity here. This is anterior, this is posterior. So wingless lies anterior to the compartment boundary, and engrailed lies posterior to the compartment boundary. Now you can imagine, now the cells that here will probably not be the same as cells that are here. Not probably, they're not the same. So this establishes the first step of identity, which is anterior and posterior. Now once this is done, then that's it. Then now the signaling pathways have to do the rest of the job, because now wingless and engrayed take over, and how the cells interpret. Now these are signaling molecules. We know what signaling molecules can do is that if it's a ligand, it can diffuse, and however it is sensed and where it is sensed, it will respond. The cells, it has the ability to affect cells that are distant from where the source of origin of the signal is. So this is because now the question is, the wingless expressing cells have to inform the neighboring cells that they are wingless, and they will form some component that the adjoining cells have to respect. And the engrailed positive cells have to inform the others that they are engrailed positive, and they're going to form structures that have to be respected. So the wingless expressing cells should not take over the job of the engrailed expressing cells, and the engrailed expressing cells should not take over the job of the wingless expressing cells. So there is mutual respect for what they're going to be undertaking. Now, how is this mutual respect achieved? Because once this mutual respect is achieved, the cells, however, even if they're proliferating, the ones that originate from wingless domain and the ones that originate from engrailed domain will know what they have to do. So they have been told what they have to do by starting from the compartment boundary. So wingless, I'll come to the next slide. So wingless will be secreted. So you can see here the cells which have very low even skipped and very low Fushitarazu, which are the pair rule genes, are going to be the wingless expressing cells. Whereas the cells which have the highest Fushitarazu expression are going to be engrailed positive cells. So this will set up the first few group of cells which will become wingless and engrailed. This is nothing new for the animal because this is what it has been doing all throughout. But now what these guys will do is what the animal hasn't done in the previous few rounds, is what is the information pass over to the other cells. Because now what will happen is that wingless, being a ligand, will be diffused, will be secreted, and it will diffuse to the other cells. Wingless will not see only cells of this side, but it's not, the ligand can diffuse between the compartment boundaries. So it will be sensed by cells on both sides of wingless producing cell. Now, when wingless is sensed by the engrailed producing cells through its receptor, it will activate the transcription of engrailed. So this kind of further reinforces engrailed expression. So if wingless is lost, engrailed expression will be reduced. Even though Fushitarazu will be there, but once the pair rule genes are gone, this wingless expression kind of keeps it going, and the compartment boundaries, and this compartment boundary will be respected. You can imagine that the pair rule gene expression over time will be lost. But if Fushitarazu was the only way to maintain engrailed expression, then once the gap genes are gone, and the pair rule genes are gone, the identity of this cell would have been lost. So that's not right. So we don't want to lose identity. So how do we maintain identity even when my even after I have lost what controls the expression of engrailed, is to put the system on an auto-regulatory loop, So the wingless expression cells seen by the neighboring cells will activate will activate the transcription of transcription of engrailed. So this engrailed is now under the control of wingless. And what engrailed does, engrailed is another transcription factor. And so the other target gene from this is the expression of hedgehog. So what happens is that engrailed producing cells will also make hedgehog. And engrailed is a transcription factor that will lead to hedgehog activation. So hedgehog is a signaling ligand, will be released by these cells, the engrailed producing cells. It will bind, hedgehog will bind to its receptor, smoothened, and then result in wingless. So this is kind of an auto-regulatory loop which maintains the expression of wingless here and hedgehog here and engrailed here. So as long as, so this guy's release is necessary for the subsequent cells to become wingless. So it's not that I will maintain myself. If I could maintain myself, then I will lose what the surrounding cells will become. But in this way, I also inform my surrounding cells to become, so in a way, these guys are also informing their neighboring cell of what their identity is. So I'm a hedgehog signaling cell, and this is a wingless signaling cell next to me. And this guy will say I'm a wingless producing cell next to a hedgehog. So this kind of a mechanism keeps two different cell types and maintains a boundary. As a result, there's going to be a sharp boundary which will be created between these two cell populations. So now, how does the sharp boundary help in defining structures within this? So this, I mean, in the screen, the denticle belts, so these are all the different bristle phenotypes that were seen, were actually, so understanding of these different hair bristle patterns that were seen were the phenotypes here was actually an outcome of the segment polarity genes. So you can see here, the wingless producing cell and a hedgehog producing cell, and their gradients. Again, now so far we did not see the establishment of compartment boundary in the previous rounds of gap and the pair rule gene-based development. Now, with the establishment of compartment boundaries, and now the utilization of gradient, the information that is going to be read. So you can see there is a declining gradient of hedgehog and a declining gradient of wingless. However, wingless is going to be interpreted, is going to give rise to the patterns that are seen here. Yeah, I mean, all of this is more detail now. I don't know if I want to go into all of these detail, but yes, I mean, this is, I would rather ask everyone to read it or go over if you want to understand how each of these bristle patterns come up. Because what is interesting is that the readout of hedgehog and wingless gradient will result in activation and repression of different signaling pathways and the utilization of different signaling pathways and different transcription factors. So now the entire, what the MPO has been doing, utilizing gradients, utilizing transcription factors, utilizing transcriptional hierarchy. All of these events are going to be now set in place to give rise to these different patterning events and giving identity to each of the cell. So yeah, so hedgehog signaling cells will activate EGFR signaling. EGFR-based signaling will result in some of these kind of bristle patterns, whereas the EGFR negative, so Rho is an EGFR ligand. And yeah, so depending on how wherever the cells are seeing hedgehog signaling, EGFR will be there. You will see bristles, and wherever cells are not seeing hedgehog signaling, EGFR is not there, and that's going to be the naked cuticle. So this is cuticle, this is naked cuticle. So essentially, these cells which have all the bristles are seeing EGFR, and as a result have bristles. Wherever it is not seeing EGFR signaling, they don't see, they form a naked cuticle. So which means all of these cells are also seeing hedgehog and hedgehog signaling, and as a result they have EGFR signaling, and these are perhaps seeing wingless and not hedgehog signaling, which is naked cuticle. So wingless, so simple establishment of compartment boundaries is giving rise to just the now utilization of the concept of compartment boundary has set up this kind of a patterning event. So that is the main point that I would like to make here, how the division of the two different cells, one and not interfering with each other's function, has given rise to this kind of a patterning event. Now, finally, the last bit is the identity. This is just about the bristle pattern phenotype, but once the segment polarity genes have been established, they will tell each segment there is an anterior and a posterior. That is what the segment polarity genes are going to be informing each segment about. Now, what the pair rule genes, gap genes, and the segment polarity genes together are going to do is in each of these domains is going to actually result in the expression of the homeotic genes. The homeotic genes are going to be essential. I mean, they're the most fascinating ones that have caught attention because they can actually cause transformation. So the homeotic genes, their expression is going to be actually determining what they're going to be. So the homeotic clusters are the antennapedia and the bithorax complex. So antennapedia complex has a set of homeotic genes, and the bithorax has ultrabithorax, abdominal A, abdominal B. This has antennapedia, labyrinth, and whatever defective labial and sex chrome reduced and deformed. So these are the cluster has these three different homeotic genes, and this bithorax complex has these three different homeotic genes. So abdominal A, B, and UBX. Now, the most classical one is if antennapedia is expressed in the head. So instead of having, so this particular thoracic segment where antennapedia is expressed is generally giving rise to legs. So what antennapedia does here, so I'm just giving an example of what they can do. So antennapedia here will repress the formation of other structures, but promote the formation of legs. Now, wherever you can choose, wherever you want to express antennapedia, Now, instead of the structure that should be developing, you will see legs appearing in that region. So the classical one is the fly having legs instead of antenna in the region. So if antennapedia is misexpressed in the head, you can see that instead of antennal structures developing, you have two legs developing. The same is true for ultrabiothorax, that if it's misexpressed in the thoracic region, in a different thoracic region, the animal develops duplications of the structures. So meaning that they're very, very potent genes, and they not only, they are very deterministic, potent meaning they're deterministic. So which means that wherever, if antennapedia, instead of being not if it is expressed in the head structures, it can override the information that is present within those cells. So if the cells of the eye have to, or the cells of the antenna have to form an antenna because they come from the head region, that information is overridden by the misexpression of antennapedia. So they, even though they have been specified and have been determined, these homeotic genes, they can override that information and redetermine wherever they are, redetermine what they can be, what they should be depending on the homeotic gene. So in a way, they are very, they're enough, they're sufficient, their expression is what is sufficient. So once the homeotic genes have been established, the structures that develop will now be set. So the establishment of this homeotic genes along the Drosophila embryo is what has to be carefully done, because if this is now missed or messed, then you will have structures which are missing, or you will have deformative structures that will be deformed or present at regions where it should not be. So essentially, once the segment boundaries have been established by the pair rule and the gap rule genes, like the anterior and posterior segments, the compartment boundaries, this will segregate each segment, and now the interplay of these pair and gap and the segment polarity genes is going to set up the establishment of the homeotic genes. Now, once homeotic genes have been done, that's it. Then the animal is now going to be, right. So once the homeotic genes are done, then essentially the animal knows what structures each of the segments have to give rise to. So that's all what I have for the Drosophila development bit. So if there are, essentially I am kind of done with my series of classes that I have. So if there are questions, we can take it up next class, or next class, or however we can, if there are things that you want me to pick up and discuss, maybe next class you can tell me. What I would want, I went over the screen paper myself. I mean, in this today's lecture, I essentially covered the screen paper also. So what I would like you all to do is to read the screen paper, and one thing is understand how they did the screen. So what were the steps involved in doing the screen? And secondly, see if there is any information that I might have missed in my class and maybe there in the paper. And the second thing is there any yeah, I mean, essentially what I would like you to read this screen paper for is to understand how they did the screen. Apart from that, what information is obtained from the screen I've probably laid out today in my today's class. So yeah, if there are questions, we can, so Friday, I think, is everybody, I mean, I would like to ask everyone if they're willing to take a class on Friday. But I mean, I don't have any other thing to talk about. If you have a particular topic that we should pick up and discuss, then let me know. And maybe that is what we can do the next class, maybe Monday or Friday. Yeah. So any questions for me today on what we spoke about, what I spoke about? Okay, no questions. Okay, fine. So yeah, so then that's it for today. Friday, I think if Friday probably we can skip a class. What I would like you to do is get back to me, either to me or to Varsha, if there are parts that you want to be taken up, and if this Drosophila, if any aspect of Drosophila development or any aspect of development is something that you would like to be hearing about or learning about, you can email me or Varsha, and then those are the things I can pick up specifically, or anything that I might have missed that you were expecting the class would cover. Yeah?
Yes.
Okay. Because I would appreciate if you do it ahead of time so that I can make some slides and or I can get the information on Monday. So I would suggest that take the time to finish the assignment, take the time to read the screen paper, and understand how the screen was done. So, I mean, I don't want you to write and submit to me and as an assignment. So it's not an assignment, but essentially for you to read how the screen was done and get your assignment for the other paper submitted, and email me if there are parts that are missing that you would like me to cover, or parts that are difficult that you would like me to speak on. Yeah, and we'll get back with, depending on that, we'll do the class. I mean, anyways, we'll have a class on Monday. I'll figure out something for Monday. Yeah, but there are any specifics, we will pick it up on Monday. Yeah. So next Friday we will take a break. Okay. I mean, not this next Friday, this Friday. Yeah.
Okay.
Okay, good. No questions for me for today, then, I understand. Okay, great. See you. Bye.
Bye.
Bye. Thank you.
Thank you.
Thank you.